Okay, so on last week, I uploaded some recorded lessons for mitosis and meiosis, and I just wanted to take a few moments this week to recap what it is that you should have gotten from that lesson. So you looked at cell division. You explored cell division independently on last week, and you took a look at both mitosis and meiosis. I wanted to make sure that we understood why cell division was important. Okay, you know that we're living things, all living things are made up of cells, right? And so the cell is the connection for the perpetuation of life. And so there's two different types of cell division that you looked at on last week. You looked at what happens during the cell cycle, particularly mitosis. And then you also looked at meiosis, right? So there are two different processes that happen for two very distinct reasons. I just wanted to take about 5 minutes today to recap that. As living things, we're all made up of cells, and so we know that our bodies are constantly replenishing our cell supply, right? Our cells don't all have an infinite lifespan, and therefore new cells must continuously be made, right? And so what happens is that these pre-existing cells that we already have in our bodies, they are going to go through the cell cycle in order to produce more cells identical to the ones that we already have. And so that process happens with mitosis. And so you looked at that last week. And I wanted to make sure that you understand the why, right? It's always important that we sort of understand the big picture here. Yes, there's a lot of fine details in terms of what goes on in living things. But we want you to understand the big picture here is that we need to produce more cells for a number of different reasons, right? In order to continue to grow and develop, we need to produce more cells. In order to repair tissue damage, new cells are being made. And also for reproduction to take place, for new organisms to be made, new cells must be made. And so therefore, mitosis and meiosis are two very critical processes in sustaining life. Right? We know that the cell is important because it contains the genetic information. That's where our DNA is housed, right? And our DNA, what we're going to look at today is heredity, right? We're going to see the connection between this DNA and how the information contained in our DNA dictates all of the things about us in terms of our, structure and function, right? And so on last week, you took a look at mitosis and meiosis, and you understand theoretically why these processes are necessary, right? Mitosis is a series of steps in which parent cell transmit genetic information to daughter cell. So this is basically the cell duplicating everything that it has. And we sometimes refer to this process as cloning, right? This takes place in all of our body cells, right? So if you didn't get that on last week, make sure you jot it down now that mitosis is the production of new daughter cells that are identical. So this happens in our body cells or our somatic cells, right? So we have two types of cells in our bodies from a general standpoint. We have our somatic cells, and then we have our sex cells. So the somatic cells are responsible for all of the growth and development and other things that go on in our body. But when it comes to reproduction, there are some special cells that we call gametes, right? These gametes are responsible for the production of new individuals, right? Our sex cells, the sperm and egg, contain 23 chromosomes. They unite together to form a zygote, and a new individual is formed. So I just wanted to just take a few moments today again to recap what you looked at on last week, making sure that you could distinguish between the purpose of mitosis and meiosis and what the outcomes are, right? So the purpose of mitosis is to make more somatic cells, right? The purpose of meiosis is simply to produce these sex cells, right? There's only one purpose for meiosis. This is strictly for reproduction, all right? From meiosis, we get our sex cells generated. Gametes are formed, right? Sperm and egg. So that's kind of the major point that we wanted you to grab from last week's lecture. We talked about the chromosomal content of humans, right? We know that we have 46 chromosomes. They exist in 23 pairs, right? The first 22 pair are our somatic, and that last pair are sex chromosomes. So I'm not going to go back through this whole thing because you did this on last week, but I want to make sure that we understood what we looked at and why, right? So we looked at the cell cycle and how, this process basically prepares the cell to be divided and make two new cells at the end of mitosis, right? So you need to be clear on, what happens during the cell cycle, right? We said the interphase is this pretty long period of preparation, right? And then we had M phase, which is where the cytoplasm actually splits off and 2 new cells are generated. All right. And we define the actual process by which the cytoplasm splits into 2 new cells and those 2 new daughter cells are created. That process is called cytokinesis. All right. If you didn't get a chance to look through that, make sure you go back and look through it. All right. And then we identified 2 basic types of reproduction. We know asexual and sexual reproduction. You should be able to distinguish between these 2 forms of reproduction and what their outcomes are. What types of organisms use these types of reproduction? Okay. We're humans. We use sexual reproduction. We know that this type of reproduction requires the union of 2 parents, right? 2 sex cells. Those gametes are being utilized here. So anyway, the last point here we'll show you is that sexual reproduction, we know that it requires 2 parents, and then the offspring that is created is basically going to be a hybrid or a mixture of both parents, right? Whereas asexual reproduction, we sometimes refer to that as cloning. It's like you're basically making an identical copy of the parent in the offspring. All right. So I'm going to stop here for that one. I just wanted to make sure that we all had a chance to get through that lesson on last week. I gave you this chart here that sort of compares these 2 processes, identifies some advantages and disadvantages of both of these types of reproduction. All right. And if you did not get through this on last week, make sure you go back and get through it because it's in your Canvas course. All right. So today we want to look at heredity. All right. There is a connection between our, this process or cell division and heredity, right? Which is the passing down of genetic information from parent to offspring. So let's take a look at that. Give me 1 second. So I want to start with this video. I think the Amoeba Sisters do a great job of connecting all of the things that we've talked about this semester in terms of our DNA and genes, chromosomes, and then this concept of heredity. And we'll take a look at how the information that's contained in our DNA is utilized to dictate those things about us from a physiological standpoint, from a physical standpoint, from a behavioral standpoint. And we'll look at some principles of genetics today and inheritance. Let's take a look at this video. You should definitely be taking notes, annotating what you feel is important. This should be a recap for you.
This foundation covers everything, but it still looks like skin. Insane.
The bases are what pair in the middle.
Oops.
The base. The week before the first day of my first year of teaching, well, over a decade ago, was an exciting time. It was also stressful because I'm a bit of a perfectionist, but a bit of a messy, unable to decorate my classroom kind of perfectionist. Actually, maybe I'm not a perfectionist. I wanted to make my classroom inviting and exciting, and I really wanted my students to walk in and think, this is amazing. I could never make it look like what I imagined up, but the most amazing thing did happen in the week to follow. I knew a colleague who had a friend who had a son who was going to college. Confusing. But anyway, that son could not take his pet snake to college with him. Some rule about dorms and snakes. My colleague asked, would you like it? They said they can give you the cage and everything. It could be a classroom pet. I didn't even have to think about it, of course. So Spike, a Texas rat snake, became a classroom pet, an amazing, popular one at that. He was friendly, well, for a snake. He'd let you hold him and not bite. That year, I was the only one in the science wing with a pet snake, and I'm pretty sure there were some people that wondered, what was the point? Well, I'm big on relevance, so anytime I would teach a biology concept, I found some way to work in Spike. Predation? Well, let's talk about Spike's appetite for rats. Mitosis? Let's talk about why Spike even needs to make more cells. One day in tutorials, a student asked me, since Spike's parents were bred in captivity, did you ever see Spike's parents? Do you think Spike's parents looked like him? You can't ask a question like that and not expect an answer. It was a great question because we were getting close to our heredity unit. Heredity is about how traits are passed down from parent to offspring. We've made a playlist of our videos that focus on heredity, including reproduction, how to track inheritance and pedigrees, how to solve genetic problems in Punnett squares, and understanding different Mendelian and non-Mendelian inheritance. But you really can't delve into those and study heredity without understanding DNA, chromosomes, genes, and traits. And that's what this introductory video is going to focus on. So back to the student question. Spike has traits. The patterns on his body, his size, these traits are coded for in his DNA. Some of the traits he inherited can be influenced by the environment. For example, if Spike had not had the nourishment he needed, that could affect his size. DNA is not just one big code buried deep down in an organism like some treasure. Spike's DNA is in the nuclei of nearly all of his body cells. He inherited his DNA from his mother and father. I can't know for sure what Spike's parents look like, but I do know that Spike inherited his DNA from them. One fun fact, many snake species can reproduce asexually. Had that been the case for Spike, he would have inherited all of his DNA from only one parent. But it would still be DNA coding for traits. Just like Spike, your DNA codes for your traits, and your cells can't function without it. DNA determines how tall you are, what color your eyes are, what color your hair is, or even if you're at risk for certain diseases. Also, like Spike, your entire DNA code is in most of your body cells. That's why in those crime-solving shows, which we may or may not absolutely love, a criminal can sometimes be caught by just leaving a cell from a hair follicle behind. DNA has a beautiful structure, and that structure will help you understand how inheritance works. DNA stands for deoxyribonucleic acid. It's a type of nucleic acid. If you remember from our biomolecules video, nucleic acids are a type of biomolecule. Nucleic acids are made up of building blocks called nucleotides. Nucleotides have 3 parts. One of them is a sugar called deoxyribose. One of them is a phosphate. We sometimes say DNA has a sugar phosphate backbone. But the most important part of the nucleotide is the base, because the sequence of the bases can actually code for traits. So as far as the bases go, there are 4 types of bases in DNA. A lot of times they'll just use the letters A, T, C, G. The A is for adenine, the T is for thymine, the C is for cytosine, and the G is for guanine. These bases actually pair in a specific way, and there's a popular mnemonic that can help you remember which of them pair together. Apples in the tree, that tells you the A for apples, T for tree, because the bases A and T go together. The other verse is car in the garage, and that can help you remember the base C always goes with the base G. Regardless of whether we're talking about Spike or a plant or a protist or a human like you, these are the DNA bases in living organisms. But the amount of DNA bases overall and the sequence of those bases we mentioned will vary among different species and also among different individuals. But it's likely to infer that Spike has a sequence of DNA bases that is more similar to his parents than he would to, say, rattlesnake. DNA has 2 strands, so there are nucleotides running up one side and there's nucleotides running up the other side. The bases are what pair in the middle. The bases are held together by hydrogen bonds. The DNA is also twisted in something we call a double helix shape. Portions of DNA make up genes. We can say, for example, that this part of the DNA here makes up a gene. Genes can code for proteins. Proteins have a huge role in expressing a trait. For example, let's consider your own eye color. Human eye color is actually pretty complex. It's determined by many genes. The genes can code for proteins involved in producing the eye color pigment, but proteins coded for by genes, they can play a wide variety of roles besides just your eye color. Proteins are involved in transport, in structure, in acting as enzymes that can make all kinds of materials, in protecting the body, and so much more. We do want to mention that not all genes are used to make protein, and there are parts of DNA that are non-coding. And even though nearly all of your body cells have your entire DNA code, Your body cells may only use certain portions of those genes. Genes can be turned on or turned off by a variety of mechanisms. We call that gene regulation. Check out our video on that. Now, you have a lot of DNA. When it's compacted, it can be organized into a unit called a chromosome. Very helpful when you're trying to make more cells and you need to get the DNA into those new cells. Chromosomes in your body involve DNA wrapped around protein structures. Humans have 46 chromosomes. That means nearly every body cell in your body has 46 chromosomes. Human sperm and egg cells, on the other hand, each contain 23 chromosomes. So you receive 23 chromosomes from your mother, 23 chromosomes from your father to give you your 46 chromosomes, your genetic code. So let's do a recap of the big picture. Here's a single chromosome. You see genes on this chromosome. These genes consist of portions of DNA. DNA is made up of nucleotides, and it's these bases here, the sequence of them, that makes the difference in coding for traits. Whew. Understanding this foundation is essential for understanding heredity, whether you're talking about you or Spike. So this may bring up more questions now, like where do dominant and recessive traits come in? What about alleles, and how do you arrange those around a Punnett square? Well, you can explore those concepts and more in our heredity playlist. Well, that's it for the Amoeba Sisters. And remember,
All right, so I thought that was a great sort of recap or reiteration of the things that we've talked about that would provide a segue into what we're going to talk about today, which is heredity and an intro to genetics, right? So you've probably heard a lot about, this concept of genes and chromosomes. We're going to take a look today and see how these genes and chromosomes play a role in determining how we function and behave. So we're going to take a look at some principles of genetics today. Oops. Share. So some principles of heredity. The goal of today's lesson, again, is not for you to become a genetic expert at the end of this lesson, but there are a few things that you should walk away with sort of a basic understanding if you were to see these terms. So the term phenotype, genotype, we're going to distinguish between those two: locus, allele, dominant alleles, recessive alleles, homozygous, heterozygous. This is going to give you some insight on how we're able to use some of these principles of genetics that we now hold to be true to sort of predict the likelihood of diseases and other traits that might be genetically based. And what we know and hold to be true, and we refer to them as Mendelian genetics, was due to the work of this scientist named Gregor Mendel. And he was responsible for, he basically conducted a lot of experiments over the years, basically breeding plants. He bred a lot of different crosses of pea plants to study what their what the offspring generations would look like. We were able to learn and elucidate a lot of information about how reproduction and genetic crossing and breeding patterns occurred. And so we refer to those principles as Mendelian genetics. And so we'll take a look at some of those today. And so just as an introduction, it's kind of what the Amoeba Sisters started to talk about at the end of that video, is that human characteristics such as our eye color and hair color are traits that are passed on from one generation to the next. So those are factors or characteristics about yourself that you had no control over. Now, obviously, you can change those or manipulate them now, but the ones that you're born with, your eye color and hair color and so forth, are due to your genetics, particularly your DNA, right? And so that's something that we want to take a moment and sort of appreciate, right? Heredity is a major part of living things. At the beginning of the semester, we identified some characteristics of living things, and one of them was heredity, the fact that you inherited genetic information from two individuals, right? Your mom and dad. So you are a hybrid of the two individuals, right? And so you may have heard individuals say to you, oh, you look like your mom or you look like your dad, because you're going to share features because you come from, the same lineage from a genetic standpoint. And so I want to first distinguish between the term heredity and genetics, right? These terms are often used interchangeably, but they're not the same thing, right? Heredity, write this down. Heredity refers to the transmission of genetic information from parent to offspring, right? So this is you inheriting DNA from your mom and dad when you're born. That's heredity, right? And we now know that this process of heredity typically follows a predictable pattern, right? If we were to do some genetic analyses on your mom and dad, we can sort of predict the likelihood of you inheriting, various traits, characteristics, diseases, and so forth, because it does happen in a sort of predictable pattern. All right. Genetics, though, on the other hand, is a branch of science. Okay. Genetics is a branch of science that studies heredity. Right. And so geneticists are those that are studying both the similarities and differences or variations within a population. Okay. And so this is very important because, again, what it does allow for us to do is kind of have some insight, and it allows us to sort of be predictable in nature. All right. Studying similarities and variations within a population from mom to offspring allows us to predict the likelihood of inheriting a disease. Therefore, we can make some preventative measures. We can do some prophylactic measures in order to prevent the onset of certain things. There's a lot of debate on the ability to have the genetic testing done, like during pregnancy. Women can opt to undergo genetic testing, and it's basically an analysis to see if your child is at risk for any, genetic abnormalities. I personally opted out of that. I did not choose to undergo genetic testing when I gave birth to my two kids because I did not want to know, right? But some people want to know. And, for me, it was kind of like, oh, no, I don't want to know, because then you're left with making some really hard choices if you find out some information you don't like. But, I had a colleague once who became pregnant, and she wanted to undergo genetic testing because she, in her words, she says, I know myself. I'm not the person who's going to be able to take care of a child who, for example, can't walk or can't talk, or, has some type of, I don't know, mental retardation, or, and so she opted to undergo the testing, and, during her pregnancy. And fortunately, she didn't have any thing that concerned her, and she continued to carry the child to term. But that's a very hard decision that you would be left with should you find out something that's less than ideal. So I think it's great that the option is there, because you do have to know yourself as a person, as a woman. I opted not to know. But anyway, so genetic testing is good. Individuals who have a family history of certain illnesses and diseases or syndromes that may be genetically based, that can be passed from parent to child, they may be more apt to undergo genetic analyses before they decide to procreate. So anyway, it's a very fascinating field of science. And so the point of this slide, though, is to distinguish between heredity and genetics, right? Heredity is the process of transmitting information from parent to offspring. Genetics is a science that studies heredity. What's going on when we pass this information from parent to offspring? We're studying similarities and variations. And variations in this case are not necessarily a bad thing. I want you to put an asterisk by the fact that variations, especially as it relates to evolution and adaptation over time, these variations sometimes provide survival advantages, okay? Sometimes they can be bad, but in some cases they're good. These changes in DNA that we see amongst populations can be advantageous. So anyway, I mentioned earlier that the things that we know and sort of hold to be true as we study inheritance was elucidated by the work of Gregor Mendel. He was a scientist who bred pea plants. right? And so what happened was he basically conducted a number of experiments over some years, collected this data, analyzed the data, and observed patterns and so forth. And now we have what we know to be Mendelian genetics. So let's start with some terminology here. As you move through this chapter, if you do reading, you watch some more videos, you're going to see these terms a lot. The gene. Okay, in genetics, this gene is referring to an inherited factor that's going to determine a characteristic or trait. All right, we're clear that this is DNA, right? So our DNA, right, segments of DNA determine, you know, specific characteristics or traits about us, right? There's a gene that codes for your eye color. There's a gene that's going to code for your skin color. There's a gene that codes for your hair color. Those are what traits are. Traits are the physical features or characteristics that we see. And so all of the genes in our bodies, they come in pairs, right? And that makes sense because if you think about it, you've seen individuals, you know, like not every trait is the same in everyone else. Some people have brown eyes, some people have blue eyes, some people have green eyes, some people have black eyes. So what, well, no black eyes, but gray, brown, so forth. And so what happens is you get 2 copies of each gene, right? Different versions of the same gene that's coding for that trait. Those different versions of the gene is called an allele, right? So if you look in the mirror and you have brown eyes, you have a dominant allele for brown eyes, right? But if we did a genetic analysis of your genes and chromosomes, we may see that you had a homozygous pair of alleles with 2 different versions where you had one allele for blue eyes and one allele for brown eyes because they exist in pairs, right? But because you have brown eyes, we are left to believe that that brown allele was the dominant allele, right? And we'll look at that concept of dominance and recessive in just a moment. But what we're understanding now is that we have genes that determine these traits, and each of these genes or traits exist in a pair of genes, if you will, that we call alleles, which are different versions or alternate forms of a gene. And so one of the traits that Mendel crossed and continued to study the outcomes was for the shape of the seed. That is something that was determined by a gene. Is the shape of this seed going to be wrinkled or round and smooth? The shape, right, which is the trait, existed as 2 different alleles. One allele encoded for the seed shape to be round, and one allele coded for the seed to be wrinkled, right? And depending on which one of these was the dominant allele or the recessive allele, that's going to dictate if we physically see a wrinkled seed or a round, smooth seed. And so all of the alleles for a particular gene are going to be found at a specific place on the chromosome. And we call that place or that location a locus. Write down the term locus. The locus is the specific place on the chromosome for a particular gene. So, you know, we have 23 pairs of chromosomes. And thanks to the Human Genome Project, we have mapped out all of the genes that are on our chromosomes. So we can go and identify exactly which gene is found on this particular chromosome, right? And the location for that gene is called the locus, right? Write down that term locus. Okay. So the point we're saying here is that there is a specific place that we'll call the locus on the chromosome in the pea plant that's going to determine the shape of that seed. All right, some other terms that we want to understand here, genotype, right? So you've heard individuals say, oh, your DNA, your genes, your set of genes that you have, the set of all of the alleles or genes that you have in your body is your genotype. It's the entire set of genes that you contain. That is your genotype. Write down that term, genotype. It is the set of alleles that you have, all of your genes in your body. Your genetic makeup is your genotype. Okay. And so when I mentioned the alleles existing in pairs earlier, right? The pairs can be identical, two identical pairs of alleles, or two different copies of these alleles. If the two alleles in that pair are identical, that is said to be a homozygous pair of alleles. Write that term down, homozygous. An organism that has two different copies of an allele, that's said to be a heterozygous pair of alleles, okay? Heterozygous pair of alleles. And so we use this information again to try and predict the outcomes of traits, right? And so let's look at this example we have here. This is a very relatable example with eye color, right? This individual A has a heterozygous pair of alleles, right? And this key tells us that the allele for blue eyes is recessive and the allele for brown eyes is dominant. I know we haven't talked about dominant and recessive in great detail yet, but stick a pin in this note that dominant alleles are going to be the allele that is manifested in a physical sense. And the recessive allele is the one that you won't see if you were to look at this individual for this particular trait. So in this case, if we were looking at individual A, we would expect that individual A would have brown eyes, right? Even though this person has a heterozygous pair of alleles for eye color, a brown allele and a blue allele, but because this blue allele is recessive, the brown allele is dominant, and so this individual would have brown eyes. We would predict that. Individual B would have brown eyes for two reasons. One being that this is a homozygous pair of alleles, so he doesn't have any other option but to have brown eyes, right? Because they're identical copies of each other. But also because brown is the dominant allele in this pair, this individual will have brown eyes. Okay, so it is important that you sort of understand this concept of alleles existing in pairs. There are different versions. So you can have, a heterozygous pair of alleles where the possibility of you having one trait or the other trait is dependent upon which trait is the dominant form of whatever which allele is the dominant form of that trait. So that's the genotype. The genotype is the set of genes that you have in your body. Then you have what we call the phenotype, right? The phenotype is your outward appearance of characteristics. So this is what you look like physically, okay? This is your physical features. This is how you're functioning from a physiological standpoint. This is your behavior. These are all the features that we're able to see from sort of an outward manifestation. And your phenotype is determined by your genotype. Write that down. This is the point we want you to understand here, that in genetics, this concept of inheritance, you're inheriting this genetic information, which is your DNA. Your genotype is a set of genes that are going to dictate your phenotype, okay? The outward manifestation of the genes that you inherit. So write it down. A phenotype is determined from a genotype. Your genotype is a set of genes that will determine what you look like and how you function and how you behave. What questions do we have? Okay. So the important concept that I want to point out to you right now as we're thinking about inheritance and this passing down of information from parent to offspring is that only the alleles or the genes themselves of the genotype are inherited. Organisms do not transmit their phenotype to the next generation, right? So you don't inherit a phenotype, you inherit a genotype. And it is the genotype that dictates the phenotype. So make sure you understand that distinction. You don't inherit traits and looks per se, but you do inherit genes that influence the phenotype. Does that make sense? Yes. Okay. Now, this concept of a dominant and recessive allele. Okay, we're almost done. Dominant and recessive alleles. Dominant alleles is a dominant allele is the form of the trait that is going to be observed in the phenotype. So this is the form of that trait that you would actually see. So we saw the pair of alleles earlier for eye color, where individual A had a blue allele and a brown allele. And we predicted that that individual would have brown eyes because the allele for brown eyes was dominant in that pair, right? So the dominant allele is the form of the trait that is going to be observed in the phenotype. This is what we would physically see. Now, on the other hand, the recessive allele is the form of the trait that is masked or repressed. We use the term repressed when we're talking about gene expression. So this form of the allele you will not see in the individual from a physical standpoint. But again, if we went back and did genetic analyses on the DNA, we could see that that allele was present. Okay. And so write this down. The only time we see the recessive form of the allele in the phenotype is in the absence of the dominant allele. So what does that mean? That means this recessive trait has to exist in a homozygous pair of alleles. It has to be two copies of a recessive allele in order to actually see that trait in the offspring. Let's look at an example and then we'll wrap up. So the important concept here is that when two alleles of a genotype are different, okay, two different alleles in that pair, we call that a heterozygous pair. When two alleles of a genotype are different, only the trait that's encoded by the dominant allele is observed. Let's look at our eye color example here. Individual A, this is a heterozygous pair of alleles. One is dominant, one is recessive. The legend tells us that the allele for blue eyes is recessive and the allele for brown eyes is dominant. So what is the probable eye color for individual A? Based on what we just learned. Someone. What would we predict to be the eye color for individual A?
Brown.
Brown. Why, Xavier?
Because it's the dominant.
Right, because the brown allele in this case is the dominant form. Thank you. Individual B, what is the probable eye color for individual B? Individual B, what's the probable eye color here? Anyone?
Brown.
Brown. It is going to be brown because we have a homozygous pair of alleles here where this person has no other option but to have brown eyes. Now, in this case, individual C is going to have blue eyes. Now, blue is recessive, and we clearly said that the only time we are going to see the recessive form of the trait in the phenotype is in the absence of the dominant form. Okay, so because both of these are recessive, in this case, this individual would in fact have blue eyes. Are we all together on this? All right, so we'll stop right here. I'm going to